All right, everyone, get ready because today we are going deep, deep, deep into antimicrobial activity assays.
Ooh, that's a mouthful.
It is, but it's really fascinating stuff. And, it's not just for like the hardcore science nerds out there.
Yeah, it's crucial stuff for anyone who wants to understand, like, how new drugs are developed, how we fight those nasty infections.
Exactly. And especially for those of you who are prepping for those BSc Biotechnology Semester VI exams. This is like prime material for that Pharmaceutical Biotechnology section.
Oh, yeah, this is going to be on the exam for sure.
So let's imagine you're facing a real brain buster of an exam question. And it asks, like, how do scientists actually figure out how well an antimicrobial works?
Right. It's not like we can just ask the bacteria, hey, are you feeling any better?
Exactly. So that's where these microbial assays come in. They're like our secret weapon to measure how effective those antimicrobials really are.
Yeah, exactly. They give us a way to actually quantify the potency of an agent, whether it's an antibiotic, an antifungal, or any other substance that can, inhibit or even kill those microscopic troublemakers.
Okay, I'm intrigued. So we're talking about a whole toolbox of methods, right?
Yeah.
What's the big picture goal behind all these assays? What are we really trying to uncover?
At their core, all antimicrobial activity assays aim to figure out how effectively an agent can stop those microbes in their tracks or even, wipe them out completely.
Like a microbial massacre.
Kind of.
Okay.
And we need to know this for a whole bunch of reasons, for developing new drugs, understanding how resistance develops, and of course, choosing the right treatment for a patient.
Got it. So it's about figuring out how much of that antimicrobial agent is needed to deliver that final blow to those pesky microbes.
Exactly. And that's where that key concept of minimum inhibitory concentration comes in, or MIC for short.
Ah, yes, MIC, the magic number.
Exactly. Think of it as the lowest concentration of an antimicrobial that can completely stop the visible growth of a microorganism. It's like finding that sweet spot, the minimum dose needed to effectively control the infection.
Okay, so MIC isn't just some theoretical lab value.
Yeah.
It has real-world implications for patients.
Oh, absolutely. It's a cornerstone of clinical decision-making.
Wow. So no pressure on those lab folks.
Well, there's always pressure when you're dealing with those microbes.
True. So how do we actually determine this MIC, this magic number? What methods do we have in our antimicrobial fighting arsenal?
Well, let's start with a classic, the broth dilution method. It's like setting up a series of bacterial spas.
Ooh, I like where this is going.
So you have these test tubes, each filled with a nice, nutritious broth where bacteria can happily grow.
A little bacterial jacuzzi party.
Exactly. And then to each tube, you add different concentrations of your antimicrobial agent. So one tube might have a high concentration, the next one a bit lower, and so on, creating what we call a dilution series.
And concentration staircase.
Yeah, a great way to put it. And after a nice, cozy incubation period, we observe the tubes. The tube with the lowest concentration where the broth remains clear, meaning no bacterial growth, that's your MIC.
So no cloudy broth means the bacteria are taking a dirt nap.
Pretty much. It's a clear sign that the antimicrobial agent has won the battle.
That's pretty ingenious.
Mm-hmm.
But is broth dilution the perfect method for every situation?
Well, like any method, it has its pros and cons. It gives you quantitative results, meaning you get a precise number for that MIC, and it helps you figure out if the agent is actually killing the bacteria, which we call bactericidal, or just stopping them from multiplying, which is bacteriostatic.
So it's not just about whether they grow or not, but how they meet their demise.
Exactly. But broth dilution can be a bit finicky. You need to be really careful with those dilutions. And it's not the best method for all types of microorganisms.
Right, because some bacteria can be quite picky eaters.
They can be real divas, sometimes demanding specific conditions to grow.
So what happens if broth dilution isn't a good fit? What other methods do we have up our sleeves?
Well, there's a close relative called the agar dilution method. It's essentially the same concept, but instead of liquid broth, you incorporate those different antimicrobial concentrations into agar plates.
So it's like broth dilution, but on a solid foundation.
Exactly. You spread the bacteria on these plates, and the plate with the lowest concentration that shows no growth, that's your MIC.
So it's all about finding that threshold where the bacteria just can't take it anymore.
Precisely. And one of the great advantages of agar dilution is that you can test multiple bacterial strains on a single plate, which can save you a ton of time in the lab.
Multitasking with microbes. I like it.
Yeah.
But I'm guessing it's a bit more labor intensive since you're dealing with those agar plates.
Yeah, there's always a trade-off, right?
That's science for you. Okay, so we've got broth dilution for precision, agar dilution for efficiency. What other tricks do we have in our antimicrobial fighting toolkit?
Now let's talk about a method that gives you a really cool visual result, the disk diffusion method, also known as the Kirby-Bauer test. You've probably seen those iconic images of agar plates with little discs and those clear zones around
Oh
them.
yeah, those are like the glamour shots of microbiology.
Exactly. So here's how it works. You create a bacterial lawn, a nice even layer of bacteria on an agar plate, and then you place these tiny discs, each soaked with a specific antimicrobial, onto the agar surface.
It's like we're deploying tiny antimicrobial bombs.
I love that analogy. As the antimicrobial diffuses from the disc, it creates a concentration gradient around it. And if the bacteria are susceptible to the antimicrobial, you'll see a clear zone of inhibition, a halo around the disc where no bacteria dare to grow.
So the bigger the halo, the more potent the antimicrobial.
Exactly. It's like a bacterial bullseye. The bigger the clear zone, the better the antimicrobials aim.
I love that analogy.
Yeah.
So this method is all about getting a visual assessment of how sensitive the bacteria are to the antimicrobial.
Precisely. It's a great method for getting quick results. It's simple, it's cost-effective, and it's widely used in labs. But it's important to remember that the results are more qualitative. It tells you if the bacteria are susceptible, intermediate, or resistant. But unlike those dilution methods, it doesn't give you a precise MIC number.
Got it. So it's more about getting a general idea of how those bacteria are feeling about the antimicrobial.
Yeah, are they cowering in fear or standing their ground?
Exactly. Okay, so we've got broth dilution for precision, agar dilution for efficiency, and disc diffusion for a quick visual assessment. But what if we want to take things a step further? What if we want to actually see those antimicrobial forces in action to see how quickly they're wiping out those bacterial bad guys?
Ah, you're thinking like a true scientist, always wanting to see the action unfold.
Well, you know, I'm a visual learner. I need to see those microbes trembling in fear.
Well, then you're going to love this next method. It's called time-kill kinetics, and it takes things to a whole new level.
Okay, I'm ready for this.
Instead of just looking at the endpoint, like we do in those other methods, time-kill kinetics allows us to track the dynamics of bacterial death over time.
Oh, wow. So it's like watching a bacterial horror movie, but we're rooting for the antimicrobial.
Exactly. We're cheering for those bacterial bad guys to bite the dust.
So how does this bacterial thriller actually play out in the lab?
So picture this. You have your bacteria happily growing in their little bacterial playground. Then we unleash our antimicrobial weapon.
Dramatic music plays.
Exactly. Now, at different time points, we take samples from this culture and determine the number of bacteria that are still alive. And by plotting those numbers against time, we can actually visualize the killing rate.
So we can see how quickly that antimicrobial is decimating the bacterial population over time.
Exactly. It's like watching a bacterial population graph plummet.
It sounds amazing,
Ugh.
but I'm guessing this method is a bit more involved than those others we talked about.
It is a bit more demanding. It involves multiple sampling and plating steps, which makes it more labor-intensive. And analyzing the data requires a bit more mathematical finesse. But the insights you gain from it are really valuable. It's like getting a behind-the-scenes look at how those antimicrobials are working their magic.
Right, more data, more analysis, but ultimately more insights into those microbial battles. So we've got broth dilution, agar dilution, disc diffusion, and now time-kill kinetics. Are there any other methods we should add to our antimicrobial fighting toolkit?
There's one more I want to introduce you to, and it's particularly exciting for those who are interested in discovering new antimicrobial compounds from natural sources.
Oh, like from plants and stuff.
Exactly. We call it biotography.
Biotography. It sounds like something straight out of a science fiction novel.
It's pretty cool. So
Yeah.
imagine you have a complex mixture, like an extract from a plant or a microbial culture,
Okay.
and you use a technique like chromatography to separate the different compounds in that mixture. It's like untangling a jumbled mess of colorful threads.
I'm visualizing a beautiful rainbow of compound.
Exactly.
Oh.
And then you introduce bacteria onto the separated mix. And after incubation, you look for those clear zones where the bacteria couldn't grow. Those clear zones correspond to the separated compounds that have antimicrobial activity.
So it's like a bacterial treasure hunt.
I love that analogy. You separate all the components, and then the bacteria tell you which ones are those potent antimicrobial gems.
That's so cool. But I'm guessing this method requires some specialized equipment.
It does. You need some fancy chromatography equipment for that initial separation step. And interpreting the results can be a bit tricky, especially when you're dealing with mixtures that contain tons of different compounds.
Right, because sometimes those mixtures can be a real bacterial battle royal.
Exactly. It can be hard to tell who's winning the fight.
So we've covered a range of methods, from the tried and true broth dilution to the more adventurous bioautography. It seems like each method has its own strengths and weaknesses, like a team of superheroes with their own special powers.
I love that analogy. And choosing the right method really depends on your specific goals and what you're trying to achieve.
Right. It's about picking the right tool for the job.
Exactly. And understanding all these methods is crucial for anyone working with antimicrobial agents, whether you're in a research lab or a clinical setting.
Absolutely. It's all about understanding those microbial battles and how we can win the war against those tiny but mighty foes.
Well said. And speaking of clinical settings, we've been dancing around this concept of MIC, that magic number that tells us the minimum concentration needed to stop microbial growth.
Right, MIC, it's not just some abstract lab value. It's a crucial piece of information that helps guide those real-world treatment decisions.
Exactly. So let's take a deeper dive into the world of MIC and its significance in healthcare, shall we?
Let's do it. MIC, here we come.
Okay, so let's really break down how this MIC, this minimum inhibitory concentration, actually plays out in a real-world situation. Like when a doctor is trying to figure out how to treat a patient.
Yeah, because it's not like they have, a microscopic ruler to measure out exactly how much antibiotic to use.
Right, exactly. So imagine a patient comes in, they have a bacterial infection, and the doctor needs to figure out, okay, what specific strain of bacteria is causing this problem?
Right.
And then what antibiotic is going to be the most effective weapon to use against that particular bacteria?
It's like a bacterial battle plan.
It really is. And that's where MIC testing comes in.
So how does it work?
So by determining the MIC of, various antibiotics against the bacteria that's causing the infection, the doctor can choose the antibiotic that's most likely to knock it out, and they can use the lowest possible dose to do that.
So it's about finding that sweet spot, the right antibiotic at the right dose to really knock out those bacteria.
Exactly. And that helps ensure, the best outcome for the patient. It minimizes the risk of side effects.
Right, because nobody wants those.
Nobody wants side effects. And it also helps slow down the development of antibiotic resistance, which is a huge concern these days.
Yeah, it's like those bacteria are constantly evolving, trying to outsmart us.
They're like tiny little masterminds.
They are. And so it sounds like MIC is a key part of our strategy to stay ahead of them.
It really is. By monitoring MIC values over time, we get really valuable information on how those bacteria are changing, how they're adapting to our antibiotics.
So it's like we're spying on the enemy, trying to anticipate their next move.
Exactly. And that helps us track resistance patterns, which is super important for public health surveillance and for developing new strategies to combat these ever-evolving threats.
It's amazing to think that this tiny little number, MIC, can have such a big impact on healthcare.
It really is. It's like a small but mighty warrior.
So how do we actually go about determining this MIC value? I know we talked about broth dilution, but is there anything else?
Yeah, so there's a variation of broth dilution that's commonly used to pinpoint the MIC. It's called broth microdilution, and it's a very precise method.
Okay, walk me through it.
All right, so first, you create a series of dilutions of the antimicrobial agents, like a staircase of decreasing concentrations.
It's a concentration obstacle course.
I like that. Then you add a standardized amount of bacteria to each dilution.
So it's like sending in a bacterial army to face off against the antimicrobial.
Exactly. Then you let them dupe it out. You incubate those tubes or wells under controlled conditions, give them a little time to interact.
Little battle arena.
Exactly. And then after the incubation period, you examine the tubes. You're looking for turbidity, which basically means cloudiness. If it's cloudy, that means the bacteria are growing. If it's clear, that means the antimicrobial is winning the battle.
So clear means victory for the antimicrobial.
Exactly. And the MIC is the lowest concentration where you see no turbidity.
It's like that critical point where the tide turns.
Exactly. It's all about precision, and every step of the process needs to be done very carefully to ensure accurate results.
Makes sense. We don't want to give those bacteria an unfair advantage.
Right. We need to be strategic.
Yeah.
Now, we've been talking a lot about broth microdilution, but there's another method that's also quite popular. It's called agar dilution.
Ah, yes, the one with the plates.
Exactly. So instead of using liquid broth, you incorporate the antimicrobial agent directly into the agar plates.
So it's like building a fortress out of antimicrobial.
A great way to put it. Each plate contains a different concentration of the agent, so you have that concentration gradient. And instead of adding bacteria to individual tubes, you spread them evenly across the surface of each plate, creating what we call a bacterial lawn.
It's like a massive bacterial invasion.
Exactly. And after incubation, you examine the plates for bacterial growth.
So we're looking for those clear zones again.
Exactly. The MIC is the concentration on the plate where you see no visible bacterial colonies.
So it's like finding that point where the bacteria just can't penetrate the antimicrobial barrier.
Precisely. Both broth and agar dilution methods are widely used. Each has its own strengths and weaknesses.
It's fascinating how scientists have developed all these different techniques.
It's all about finding the best way to understand those microbial interactions. Now, there are actually a few other methods besides these two mainstays that can be used to determine MIC.
Oh, really? Tell me more.
So one method is called the gradient strip method. It uses a strip with a preformed gradient of antibiotic concentrations.
It's like a mini concentration staircase.
Exactly.
Yeah.
You place this strip on an agar plate that's been inoculated with bacteria, and the MIC is determined by visually identifying the point where bacterial growth is inhibited.
So it's a visual representation of the MIC.
Precisely. It's a relatively simple method to perform, but it's important to remember that the accuracy can be affected by things like how quickly the antibiotic diffuses through the agar.
Right. Those antibiotics can be sneaky sometimes.
They can. And then there are also automated systems that use broth microdilution techniques to determine MIC values very quickly and accurately.
Robots doing microbiology? That's amazing.
It's incredible how technology is changing the field. But it's important to remember that even MIC testing has its limitations.
Oh, really?
Mm.
It's not a perfect predictor.
Exactly. The MIC value is determined in a controlled laboratory environment, which doesn't always perfectly mimic the complex conditions inside a patient's body.
So it's like comparing apples and oranges.
In a way, yes. There are so many factors that can influence how well an antimicrobial actually works in a real-world setting.
Like the patient's immune system, for example.
Exactly. The immune system plays a huge role. And then there's the site of infection. Different parts of the body can be more or less hospitable to certain antibiotics.
And what about the drug itself?
The drug's pharmacokinetic properties, how it's absorbed, distributed, metabolized, and eliminated by the body, all of these things can affect how well it works.
So MIC is a valuable piece of information, but it's not the whole story.
Exactly. It's one piece of the puzzle, and clinicians need to consider all the other factors, too.
It's about using MIC as a guide, not a rigid rule.
Exactly. Now, we've been diving deep into MIC, but I'm also curious about those other agar diffusion assays we talked about earlier.
Yeah, we touched on the disk diffusion method. But are there other ways to visualize those microbial battles on agar plates?
There are actually quite a few, and they're all really cool because they give you that visual representation of microbial activity.
Okay, I'm ready for this.
So besides the disk diffusion method, we have the, diffusion method, the agar plug diffusion method, the spot assay, and the cross-streaking method.
Wow, that's a lot of agar action. Let's start with well diffusion. How is it different from disk diffusion?
So instead of using those little disks, you create wells in the agar, and you add the antimicrobial agent directly into those wells.
Like tiny little swimming pools for antibiotics.
I like that. And just like in disk diffusion, the antimicrobial agent diffuses out from the well, creating that concentration gradient. And if the bacteria are susceptible, you'll see that clear zone of inhibition around the well.
So same principle, different delivery method.
Exactly. The main advantage of well diffusion is that you can test liquid samples directly.
So it's a bit more convenient.
Yes. But the downside is that you have to prepare those wells, which adds an extra step to the procedure.
Got it. So it's a trade-off. Okay, what about agar plug diffusion? What makes that one special?
So agar plug diffusion is really cool because it lets us study microbial interactions. Specifically, it allows us to see how one microorganism might inhibit the growth of another.
Oh, wow. So it's like microbial warfare.
It is. You have two contenders, a producer strain that secretes antimicrobial compounds and an indicator strain that we're testing for susceptibility.
Okay, so how do we set up this microbial battle?
So first you grow the producer strain on an agar plate, give it some time to produce those antimicrobial compounds. Then you carefully remove a plug of agar containing those compounds and transfer it to a fresh plate that's been inoculated with the indicator strain.
It's like planting a microbial bomb.
Exactly. And then after incubation, you look for zones of inhibition around the agar plug.
So the bigger the zone, the more effective the producer strain is at fighting off the indicator strain.
Precisely. It's a great method for screening microorganisms for the production of novel antimicrobial compounds. But it can be a bit tricky to interpret the results because the size of that zone can be influenced by a few factors, like how quickly those compounds diffuse through the agar and how sensitive the indicator strain is.
So it's not always a clear-cut victory.
Right. Sometimes those microbes can be sneaky. Now, moving on to a method that's all about speed and efficiency, the spot assay.
Spot assay. Tell me more.
So with this method, you literally spot small volumes of the antimicrobial agent directly onto an agar plate that's been inoculated with the bacteria you're testing.
It's like a microbial shooting range.
I love that.
Yeah.
It's a quick and easy way to screen a large number of samples for antimicrobial activity. You just look for those clear zones of inhibition after incubation.
So it's a great way to get a quick yes or no answer.
Exactly. It's a qualitative method. It tells you if there's activity, but it doesn't give you that precise MIC value.
Got it. So it's like a screening test to see who's got potential.
Exactly. And finally, we have the cross-streaking method.
This one sounds interesting.
So this method is all about observing how two microorganisms interact on an agar plate.
Okay, I'm picturing it.
So you streak one microorganism down the center of the plate, and then you streak the other one perpendicular to that first streak, creating a point where they intersect.
A microbial crossroads.
Exactly. And then you look at that intersection point. If the first microorganism produces antimicrobial compounds that inhibit the growth of the second one, you'll see a clear zone of inhibition.
So it's a visual way to see if they're friends or enemies.
Exactly. It's a really cool way to study those microbial interactions.
Wow, we've covered a lot of ground today, from MIC testing to all these different agar diffusion assays.
We have. It's been quite a journey through the world of antimicrobial activity testing.
And it's clear that this is a field that's constantly evolving, especially with the growing threat of antibiotic resistance.
Absolutely. We need new strategies, new approaches to discover those next-generation antimicrobial agents.
So what do you think the future holds for antimicrobial discovery? What new methods might be developed?
That's a really, really huge question, and it's something that a lot of scientists are working on right now. The methods we've talked about, they've been incredibly valuable, but this whole problem of antibiotic resistance really shows us that we need to keep innovating. We can't just stop there.
Yeah, it's like we're in this constant arms race with those microbes.
It really is. And one of the most exciting areas right now is exploring the microbial world itself. Those microbial gladiators we talked about, they're producing all these antimicrobial compounds as part of their survival strategy.
It's like a microscopic battle royale out there.
Exactly. And we can use that to our advantage. Scientists are now using some super sophisticated techniques to identify new antimicrobial compounds that are produced by, bacteria, fungi, even viruses.
So we're basically looking for those hidden gems in the microbial world.
Exactly. And we're not just looking for them, we're actually creating them too.
Wait, what do you mean creating them?
So there's this field called synthetic biology, which is basically like genetic engineering on steroids.
Okay, that sounds intense.
It is. Scientists can actually engineer microorganisms to produce new antimicrobial compounds that don't even exist in nature.
That's mind-blowing.
It is. It's like we're becoming microbial architects, designing and building these next-generation antimicrobial weapons.
So we're not just searching for new weapons, we're making our own.
Exactly. And these synthetic antimicrobials, they can be designed to be more potent, to have a broader spectrum of activity, and even to be less toxic.
So less risk of side effects.
Exactly. It's all about precision engineering at the microbial level.
That's incredible.
Yeah.
But, even beyond just finding or creating new compounds, there's this growing realization that we need to be smarter about how we use antibiotics.
Absolutely. It's not just about having more weapons, it's about using them strategically.
So what are some of those strategies?
Well, one really interesting approach is to target bacterial virulence factors.
Virulence factors. What are those?
So those are basically molecules that bacteria produce that help them cause disease.
So instead of trying to kill the bacteria outright, we're just trying to disarm them.
Exactly. And that's a huge advantage because it reduces the pressure on those bacteria to evolve and develop resistance.
Like we're outsmarting them.
Exactly. And then there's another really cool approach called phage therapy.
Phage therapy. What's that?
So phages are viruses that infect and kill bacteria.
Wait, viruses that kill bacteria. So it's like the enemy of my enemy is my friend.
Exactly. Phage therapy has actually been around for a long time, but it's gaining a lot of attention now because of this whole antibiotic resistance crisis.
So how does it work?
So you basically use these phages to target and kill specific bacteria, and they have some really amazing advantages.
Like what?
Well, first of all, they're super specific, so they usually only attack a narrow range of bacteria, which means less damage to those good bacteria that we need.
So less collateral damage.
Exactly.
Yeah.
And because they can evolve along with their bacterial hosts, it's much harder for those bacteria to develop resistance.
So it's like they're constantly one step ahead.
Exactly. And beyond these specific approaches, there's also a lot of emphasis now on understanding the bigger picture. The interaction between the bacteria, the antibiotics, and the host's immune system.
So it's not just about the bacteria in isolation.
Right. It's about understanding the whole ecosystem and how everything interacts.
It's like a complex web of relationships.
Exactly. And to really combat infectious diseases effectively, we need to understand that complexity.
Wow, this has been an incredible journey. We've learned so much about antimicrobial activity assays, from the basics to the cutting edge.
It's been a real deep dive.
And I think it's clear that this is a field that's full of exciting possibilities.
Absolutely. There's so much potential for new discoveries and new innovations.
And for those of you listening who are preparing for your pharmaceutical biotechnology exams, I hope this deep dive has given you a solid foundation.
Absolutely. These concepts are going to be super important for those exams.
But even if you're not taking an exam, I think this is just fascinating stuff to know.
It really is. Antimicrobial research affects all of us, and it's important to be informed and engaged.
Well said. So on that note, we'll wrap up this episode of the Deep Dive.
Thanks for having me.
It's been a pleasure. And remember, knowledge is power, especially when it comes to the world of microbes.
Stay curious, everyone.
Until next time.